Good morning. I'm Jerome Kim of the International Vaccine Institute, and I'd like to thank the organizers of the University of Hawaii Innovation Virtual Conference for the opportunity to speak today. I'm really sorry that I couldn't join you there in Hawaii, but as many of you realize, COVID has forced a number of changes in many of the things we do and the ways we do business. So we never expected any virus, to be fair. The late Sir Peter Medawar once called the virus a bit of bad news wrapped up in protein, and COVID-19 has proven to be successive months of bad news. As of October 27th, 2020, there were over 43 million infections, over 1 million deaths. And we know what we have to do. In order to control the virus, several countries around the world, not only in Asia, have used a combination of extensive testing, tracking, isolating infected cases, and treating infected cases in order to bring the epidemic under control, and then used at the same time a number of interventions to prevent further transmission. This would include distancing, sometimes lockdowns, masks, and hygiene. What you see on the bottom panel is actually what happens in Korea, which has a population of 51.6 million. And you can see the number of daily new cases from February until October. It's characterized by a series of suppressive episodes, followed by outbreaks, followed by further suppression. And that may very well be the now normal and the new normal until we have an effective vaccine, which will allow us to have a new weapon in the armamentarium of prevention and control that will eventually, hopefully, be able to bring this pandemic to a close. I can't emphasize enough the importance of public support. Those countries that have been successful in reducing the impact of COVID-19 on lives and economies have had the support of the people and willing compliance with government instructions. So as you approach COVID vaccine development, a number of questions naturally pop up. First, does natural COVID infection prevent reinfection? This is actually very important because vaccine pathogen combinations that are associated with, where infection is associated with prevention of reinfection, actually have a much higher likelihood of successful vaccine development. So this would apply to, for instance, hepatitis A or polio. It does not apply to HIV. Natural immunity does not control HIV infection, and hence the problems that we've seen with HIV vaccine development. The second question we're often asked is, will we need seasonal COVID-19 vaccines? In fact, we know that the amount of sequence variation in COVID-19 is 8 to 10 times lower than we find in influenza in any given year, and is thousands of times lower than what we see with HIV. So there's a hope that COVID will be more like the rotavirus vaccine, where the vaccine approved in 2007 is still effective today in preventing rotavirus diarrhea, rather than like the flu vaccines, which are altered every year. The next question is, what are the relevant protective immune responses? We think that it's probably neutralizing antibody, that is, antibody that binds to and inactivates the virus, but there's some suggestion from animal studies that it may actually be a wider class of antibodies that also serve important functions, allowing the viruses to be opsonized and digested or taken up by phagocytic cells. The next question is, are animal models predictive? We hope they are, because so far 14 non-human primate, that is monkey studies, have shown that a number of different vaccines protect against disease. The final question is, are there safety concerns? And there are some hypothetical safety concerns that applied originally to animal studies of the SARS-1 virus from 2002. So far in those 14 non-human primate studies that I previously talked about, we haven't seen any evidence of safety issues. And so far, all the studies that have been paused are now ongoing in human clinical trials. So again, we hope that there are no safety issues, but we really don't actually have that long a duration of follow-up for most of the vaccine trials. So it usually takes 5 to 10 years to develop a vaccine. It costs roughly $1 billion, and the failure rate from laboratory to licensure is about 93%. What you see on the top panel is a hypothetical 5-year vaccine development timeline. How are we going to compress that and absorb the risk of development in a time frame that is really 12 months? What you see is a hypothetical design for an accelerated vaccine development timeline. Preclinical, that is the studies that are done in animals, are done at the same time as the first studies in humans. This is allowed by regulatory agencies on one condition, that is that the vaccine or a similar vaccine, that is having the viral backbone such as chimpanzee adenovirus, has been used safely in thousands of human subjects. And that is the case for DNA, for RNA, for Ad26, and for many of the other vaccines. Phase 1 proceeds with the collection of safety data. 2 weeks after the final volunteer is vaccinated in phase 1, the study continues, but the Data and Safety Monitoring Board reviews the safety data and, if the vaccine appears safe, allows the vaccine to progress to phase 2. Phase 2 looks at the generation of the correct immune responses, that is immunogenicity and safety. And again, 2 weeks after the final vaccination in phase 2, the company analyzes the protective immune responses. Is there neutralizing antibody or not? And if the Data and Safety Monitoring Board review the data and accept it, then the study moves on to phase 3, which is the study of safety and efficacy. Now, phase 3 trials are often designed with a sneak peek, something called an interim analysis, which is actually a formal part of the analysis plan. And if the data look good enough, if there's evidence of protection, then a company may bring the product forward for emergency use approval from the US Food and Drug Administration. Now, we don't know that that will be the case. And sometimes if the data are good enough, the Data and Safety Monitoring Board halts the study for efficacy. That is to say that to continue the study would impose undue risk on the placebo recipients who are not protected. We're not anticipating that that will be the case, but at this point, of course, we don't know. I think very importantly, under the normal 5 or 10 year development cycle, we have 5 or 10 years to collect safety information. We don't have the luxury of doing that. What the US FDA has mandated instead is that the vaccine trials prior to emergency use authorization have a median of 2 months of safety follow-up. So that would cover the bulk, the vast majority of adverse events that are occurring after immunization. Well, how are you going to get a big company like Merck or Pfizer to move faster when they're looking at $1 billion cost and a 93% failure rate? Well, to accelerate, you help them to de-risk the process. And how do you de-risk? You provide money. So to de-risk, the US government has provided funding, $10.3 billion worth of funding. And you can see, for instance, with AstraZeneca, with a chimpanzee adenovirus vaccine, the US government provided $1.2 billion of support in exchange for potentially 300 million doses of vaccine. CEPI has done a similar thing, but with an order of magnitude less funding, with $1.4 billion, which is still a lot, but they've often funded many of the companies that the US government has funded, including AstraZeneca/Oxford, Johnson & Johnson, Novavax, and Inovio. This is just a list of the roughly 300 plus candidates, divided first in the first table to the left into the different types of vaccine platforms that are available, and then an actual listing of the human clinical trials on the right. There are 46 vaccines currently in human clinical trials, and the number changes weekly. This is actually slightly more helpful because it describes the front runners, those studies that are in phase 3 or soon to enter phase 3. And you can see with the little stars, those vaccines that are already approved for emergency use in particular countries. That would be China and Russia. Moderna, and you can look at the sample here, started its initial clinical trials at the first circle, the 17th of March, 2020. By the end of April 2020, they had reported the results of immunogenicity studies by press release. And then by July 27th, 2020, they started their phase 3 trial. What's shown in the hatch bars are when the companies are anticipating beginning manufacture of the vaccine in anticipation of success. So you can see that many companies, including the Chinese company Sinopharm, but also now we know Sinovac as well, have started the production of their vaccines in anticipation that safety and efficacy will be found at the interim analysis in November or December. What I show here is actually all the different vaccines that have entered phase 2/3 or phase 3 studies, divided up by their neutralizing antibody titers. Now remember, neutralizing antibody is that infection-fighting protein that binds to and inactivates viruses. You can divide it very naturally into 3 groups. The lowest group is under a titer of 100, that's in gray. The middle group in yellow has titers between 100 and 500, and the companies in green have titers associated with vaccination of over 1,000. Now, What does this mean? We think that neutralizing antibody is good, more might be better, but in fact, all of the products, except for CanSino's where there is no data, protect monkeys against COVID disease. So does neutralizing antibody really matter? We don't know, but you can see them racked and stacked here by neutralizing antibody titer. But in addition to proving a vaccine works, we're also going to have to make it, make a lot of it, 8 to 16 billion doses of vaccine. Now, we know that around the world, there's roughly 10 billion doses of vaccine manufacturing capacity. Not all of that can be used to manufacture a COVID vaccine. COVAX, which is a facility that is formed by a collaboration between WHO, CEPI, the Coalition for Epidemic Preparedness Innovations, and Gavi, the Vaccine Alliance, hopes to have 2 billion doses available by the end of 2021. Operation Warp Speed, which is the US effort, already has pre-ordered hundreds of millions of doses of COVID-19 vaccines. In fact, the number is closer to 1.5 to 1.6 billion doses. But I think very importantly, and this may be the one silver lining that comes out of all of this, that vaccine manufacturers, high-income country vaccine manufacturers like Johnson & Johnson or like AstraZeneca are partnering with developing country vaccine manufacturers to make high-quality vaccines in large volume at low cost. COVAX is central to this. So COVAX is an organization formed by 190 countries. 70 of the contracts are already in place. 90 low and middle income countries will receive the vaccine either free or at greatly reduced cost. COVAX hopes to purchase 2 billion doses of a WHO approved vaccine by the end of 2021. This reflects roughly 20% of need. That will allow you to vaccinate, for instance, the elderly and healthcare workers. Importantly, the United States and Russia have not joined COVAX. China just recently joined. COVAX aims for global access. It wants to have impact and transparency, and it wants the countries involved in COVAX to have some solidarity around a vaccine solution for the COVID-19 pandemic. So final thoughts. First, we'll likely have evidence of safety and efficacy by the end of this year or early next year. But what does the day after efficacy look like? Well, importantly, who's going to make 8 to 16 billion doses of vaccine? And second, who's going to ensure access and equity in allocation so that low and middle income countries also have access to a vaccine solution? I think it's commonly said that until everyone is safe, no one is safe. And that is the whole principle operating the COVAX mechanism. The other big questions. We didn't have time to optimize the vaccine schedule or dose. We don't know if booster doses are going to be important. So that's an important question to answer the day after efficacy. The other questions are, what are the laboratory correlates of protection? That's actually a really important question because it allows you to greatly accelerate and reduce the cost of development of the next generation, perhaps even more effective COVID-19 vaccines. Finally, what we really will lack, because all we're doing are these large randomized double-blind clinical trials, is real-world evidence. It's one thing for a vaccine to have a 60% efficacy in a trial that compares 15,000 people who got placebo to 15,000 people who got vaccine. But what's going to happen when you vaccinate 300 million people? What is going to be the level of herd immunity that we see? A really critical question because countries around the world are banking on herd immunity to protect unvaccinated people. And we don't know that any of the vaccines will generate this, and we don't know at what level of vaccination we'll have to be in order to see herd immunity. The next important questions have to do with safety and long-term safety, anticipating the forces that will oppose the institution of vaccination, the anti-vaxxers, the anti-science people, the anti-mask people who are kind of moving together, conspiracy theorists. So we're going to really have to work to anticipate this and prevent this from interfering with vaccination of significant numbers of the population. Finally, the questions around licensure and emergency use, I think, have been greatly simplified, at least in the United States, by the publication of a document by the US FDA around emergency use, in particular around COVID-19 vaccines. And then finally, the vaccine is only a weapon in the war against COVID-19. And we really are going to need to do vaccination. We're going to make an army that will carry out vaccination and allow the world to leave the COVID pandemic. Thank you very much.